Welcome to UT Brainstorms. Thank you so much for being here. I used to glance to see which Brainstorms this is, but I think we're actually in the 80s now. This is something like the 86th or 87th Brainstorms, and so many faces that have been to so many of them. It's fun to see. How many of you is this your first Brainstorms? So that's great too. So my name is Mike Mock. I'm a member of the Department of Neuroscience here at UT, and we do Brainstorms in part because outreach is important to us. We want to share our love of neuroscience with you. It's also important to us because we learn a lot from you, from talking to you, from the questions. And that's one of the reasons why so much of Brainstorms is about question and answers. And so for those of you who haven't been here before, our format is pretty simple. We'll have about a 35 or 40-minute presentation just to kind of prime the pump and get us going. And then we'll do question and answers with our speaker and the two panelists until it's time to leave. All right? We have two ways of submitting questions. You can some people prefer to raise their hand. We'll have people running around with microphones. We have this online site where you can submit questions. You can submit questions now. You can do it during the presentation. And just to keep things snappy, we'll go back and forth between the two of them. All right. Next announcement is parking. We have parking vouchers here. This is good for Speedway and for San Jac. We always recommend Speedway because we've had a lot of trouble. San Jac, one time, was it last time? There was a concert there, and so they didn't accept these. But barring any trouble at San Jac, this will work for both. Just grab one on the way out, and then we don't have to deal with parking. Lastly, the final Brainstorms of this season will be Thursday, May 21st. Same room, same time. I usually end the season just because it's easier to do that than to find a colleague of mine who wants to do it that late in May after the semester's over. And by popular demand, this will be about the fourth or fifth one, but we'll talk about dementia and things that are near and dear to us. So that's what's up with what's coming. Thank you for being here. It's my pleasure. Distinct pleasure to introduce our speaker tonight, Dr. Laura Colgin. She's one of our superstars. I came here 19 years ago in part to help hire people to build neuroscience at UT. Laura was was in this amazing wave of hires that we had in that era, and all of them have become superstars. Laura is an internationally recognized neuroscientist and tells a great story, so it's a great treat to have her here tonight. And she'll do her thing. She'll introduce our two panelists, and then we'll do lots of questions and answers, okay? Laura, thank you so much.
Oh, thank you. Thank you, everybody, for coming out. It's just great to see so many people interested in neuroscience, which is a topic that is really our favorite thing in the whole world. So it's wonderful to see you all. And thank you, Mike, for the kind introduction. So my lab studies a variety of different things, but I think the topic that we are best known for is, is rhythms in the brain. It's also the topic of the undergraduate course that I teach here, an upper division course that I teach here at UT Austin. And I'm going to be telling you about the importance of rhythms in the brain today. So just to start out, I know we have a general audience, different varying levels of knowledge. I'm sure some of you have heard this before, especially if you've been regular Brainstorms attendees. But I just want to talk a little bit about how our brains code information. So, for example, if you are making a memory, you're storing a memory of this experience that you're having right now, how is your brain doing that, and how is it going to retrieve it later? Well, in the human brain, we have about 100 billion brain cells, and we call those cells neurons. That's a specialized type of cell in the brain that transmits information. And so each of these neurons is going to send a particular message related to your thoughts, your actions, your perceptions of the world around you, or your memories that you're currently storing of this experience, or maybe that you are recalling of the last time that you attended an event like this. So the way that neurons send these messages is they transmit signals by actually emitting little spikes of electricity. And they send these little spikes from one neuron to the other. And if the spikes are frequently sent from one neuron to the next, then the connection between those neurons will be strengthened, and those neurons will be potentially part of the same memory representation that's stored. Now, we're not in normal, healthy humans that are just, going about our daily lives and not undergoing any sort of surgery or brain surgery or anything like that. We don't have a way to record these spikes, these electrical spikes from individual neurons, but there is a way to measure what your brain is processing at a given time, and one of the most powerful tools for doing that is this tool called the EEG, or electroencephalogram. And Hans Berger discovered the electroencephalogram in the early 1900s. It's one of the most important developments in clinical neurology. And the way that he discovered this is actually quite interesting. He was part of the cavalry in World War I, and he had a really bad horse riding accident. And at the same time that he had that horse riding accident, his sister had a feeling, a premonition that something really bad had happened to him. And so he thought, wow, is there a way that our brain is giving off signals that can be picked up by other people? Now, of course, today we don't believe that to be the case with what we know about physics and things like that. But this led him to say, well, let me put some electrodes on the surface of the brain that can potentially pick up electrical activity and see if anything is coming out of the brain. And when he did that, he discovered this rhythm right here. Here you can see an unfiltered signal from one of those electrodes, and this is a rhythm known as the alpha rhythm. It has a frequency of about 12 cycles or 12 waves happening per second. This is the unfiltered signal, and below is the filtered signal. And so then those of us who study brain rhythms, we're off to the races. And if you have ever had your own EEG measured, or you've ever played one of those games where you have the headset that measures your EEG and you try to concentrate and move a ball around a screen or do something like that, this is the rhythm that's the easiest to elicit. And that's because it comes on very strongly when you relax or when you close your eyes. And we'll talk about why that is the case in a second. So what exactly is the EEG measuring? Well, as I mentioned, this is a non-invasive technique. This doesn't require surgery. It just requires sticking an electrode to the skull. And so what that means is that any electrical activity that's being generated within inside your brain has to go through all these layers of material that are not particularly well suited for conducting electricity, like the skull, your skin, hair, things of that nature. So what that means is that only the largest activity signals that are being generated in your brain are going to be able to be picked up with this technique. And the largest signals that are going to be detected are going to be neural activity that's synchronized across a lot of different brain cells. So the analogy that I like to use for this is, have any of you, and especially we have Dr. Mock here, who is a baseball player, so we're going to use this baseball analogy. He was actually a collegiate athlete playing baseball. So have any of you seen that movie Moneyball with Brad Pitt? Yeah, it's a really good movie, right? So he's the manager of the Oakland A's, and he gets really nervous when the team is playing. So he can't be out on the field. He has to be behind this concrete wall. He's not watching what's going on. So you can imagine if you have a bunch of people out on the baseball field or in a stadium, and you're behind a concrete wall, and all the individual people are having their own little individual conversations. They're all transmitting independent information. You're not going to hear anything, right? It's just going to be background noise. But if everybody starts chanting in unison, hook 'em horns, like everyone's yelling that, thousands of people yelling that at the same time, you will be able to hear it beyond that brick wall, right? So the concept of EEG is the same. If thousands of neurons are engaged in the exact same activity at the exact same time, then the EEG will be able to pick it up quite nicely. And that's why the EEG is really well suited for picking up brain rhythms, which reflect synchronized activity across large groups of neurons. Okay, so I'm using a lot of jargony terms here. What do I mean when I talk about synchronous activity across different brain cells? Just means activity that is occurring at the same time. So I'm going to show you in this little cartoon, if you can imagine, these are, a bunch of different brain cells, and you can imagine that they're all being activated at slightly different times. So if all of that is going to sum together, that's like the people out on the baseball field that are all having their own individual conversations. They're not in sync with each other. And so the output is just going to be very small. It's not going to be able to be picked up by the EEG. But if in contrast, all of those neurons are activated at the exact same time, then they are going to generate a large and powerful output that's going to make its way through all that resistive material, the skull, the skin, the hair, et cetera, and get picked up nicely by that EEG electrode. Okay, so when activity occurs in sync across lots of different cells, periodically, meaning that it's happening over and over in a repetitive way, then that is going to generate EEG rhythms or brain rhythms like you can see here. And rhythms are going to have a phase or a portion of each wave that is more excitatory, and another portion where things are going to be more quiet. So here you can see all the cells are active, and then they all take a break, and all the cells are active, and then they all take a break, and so on and so forth. So this provides a mechanism for the brain to actually coordinate the activity of large populations of cells that are engaged in the same function, such as coding different types of information that is going to be part of the same coherent memory. Now, in our brains, we have lots of different brain rhythms, and they all have different timing patterns, which in the brain rhythms field we call frequencies. You can also think of this as being related to its wavelength, or you can just think we have slower rhythms and we have faster rhythms. And so here, this is an example of a slow rhythm. This is a delta rhythm. This occurs with one cycle, one wave per second. And this is a rhythm that is going to be dominating your brain when you are in the deepest stages of sleep, slow wave sleep. This is another type of rhythm, the theta rhythm. This is the rhythm I'm going to be talking the most about today. This has a frequency of approximately 5 cycles per second or 5 waves per second. And this is a rhythm that comes on very strongly in certain brain areas when you are paying attention, when you're learning, when you're actively engaging with the world around you. And also, mysteriously, during the REM stage of sleep, which is when we are dreaming. Here is our friend, the alpha rhythm. As I mentioned, this was the very first brain rhythm that was discovered by Hans Berger. You can see another picture of it here. And as I mentioned, this is a rhythm that dominates when you close your eyes, when you're relaxing, when you're not engaging with sensory information in the world around you. So this is a rhythm that is actually known to have a suppressive action on cells and make sure that they're not actively transmitting information. So you can think about this coming on in the areas of your brain that process visual stimuli in the world around you, visual information, because if you close your eyes, there is going to be a little bit of light that's still making its way into your retina, which is the part of your eye that's going to transmit visual information into your brain. But you have your eyes closed, so any thing that's getting activated is actually meaningless. because your eyes are closed, right? So this rhythm would come on very strong and suppress everything to make sure that any light that gets through, you're not trying to make sense of it because you're not actively taking in visual information. You're just going to suppress that. It's just irrelevant. And there's other rhythms here that I'm going to skip. We're not going to talk about those ones today. But the point is, there's lots of different types of brain rhythms, and they all have different functions. And if you have questions about some of these other brain rhythms, I'm happy to talk about them in the Q&A. Okay, so why does the brain use different frequencies of rhythms? Well, there's a couple different reasons about it for the brain to use different frequencies of rhythms, but the main one that I'm going to talk about is that the timing of the frequency of brain rhythms actually matches their functions. And the example that we're going to use is the rhythm that my lab is probably most often studying. It's probably my favorite brain rhythm, although it's hard. It's like probably choosing a favorite child or something, right? I love all the brain rhythms, but I really do love the theta rhythm. And this is a rhythm that is really strong and really dominates in the main brain area that my lab studies, which is called the hippocampus. This is a key brain area for memory. Some of you may have heard about it before. And this is the main brain rhythm that is associated with the hippocampus when you are engaged in active behaviors, like actively exploring the world around you, actively taking in information from the external environment, paying attention, forming a new memory of what you're experiencing. This is the rhythm that is going to be dominant in the hippocampus. And this is an example recording of theta rhythms down here. This is from a rat. I'll come back to that in a second. This is the primary research model that my lab uses. But these rhythms are not recorded as beautifully in humans, but they do occur in the human hippocampus as well. Okay. So I said the timing of these brain rhythms matches their function. And when you think about this with theta rhythms, I said these rhythms are coming on, they're dominating the hippocampus when you're taking in information from the world around you. And the timing of this rhythm actually matches the motor actions that we engage in to take in information from the world around us, and also that animals engage in to take in information. So in animals that use sniffing or olfaction as a primary sense, like a dog or a rat, or in this picture, a deer, when they are actively sniffing and sampling an odor, that sniffing is occurring at theta frequency. And so if I were to play theta rhythms for you, it is in the range of your conscious perception. You would be able to distinguish the individual cycles. Now, there's also animals like rats and mice, cats, that use whiskers to take in touch information, which as neuroscientists we call that somatosensory information. And what's really interesting is when these animals move their whiskers, if they're just randomly moving their whiskers in air and there's nothing to touch, they don't do it at theta frequency. But if they go up to something that they want to see what's the texture of it, they want to get some information about it, then they will start whisking at theta frequency. Also, are there any students in the class that get your lectures recorded and then play them on 2 times as fast? Don't do that, please. Because theta rhythms have been known since the 1970s to be very important for learning and memory. And the rhythm of syllables in our speech matches the timing of theta rhythms in the human brain. So if you are speeding up your lectures, you're making it hard for your brain to take in that information into your hippocampus and for you to learn it and retain it long term. So then you might take your upper division course and your professor might say, do you remember what you learned back sophomore year? And you're like, no, actually I don't. So just let's live life, and our brains have evolved to live life in real-time speed, right? If you're listening to an audiobook or something you don't need to remember, then that's fine. But maybe for your classes, just please, real life speed. Okay, and then also as humans, many humans use vision as one of our dominant senses. We're always taking in information from using our eyes, right? And we have a part of our eye called the fovea that is the highest resolution part. So when we want to pay attention to something, we direct that part of our eye towards the thing that we really want to pay attention and take in. And so we're constantly doing these eye movements to fixate on whatever part of the visual world that we're the most interested in. And those movements are called saccadic eye movements. And the timing between saccadic eye movements also matches the frequency of theta rhythms in humans. So you see that the timing of this rhythm is really ideally suited for taking like a snapshot of the external world over and over, a new snapshot, update it, a new snapshot, and then let's take it into memory and store a memory of it for later. Okay. So what does the hippocampus actually do? This is a brain area that is particularly important for what we call episodic memory. So these are memories of episodes in your life or events in your life that you experienced, like maybe your daughter's wedding or your birthday party last year, or that time that you took a really fun fishing trip, or even things as simple as like where you put your keys or where you parked your car. So you can think the hippocampus is important for these type of memories as opposed to other types of learning, like learning how to play golf or something like that. That's not going to be your hippocampus, right? It's these autobiographical memories. And here is a picture of a human brain. And you can see this pink structure here. This is like in your medial temporal lobe here. This is the hippocampus. It's a deep-lying structure, so if you can imagine me pointing inside of my brain going that way. So what does the hippocampus do exactly? Well, there's different parts of your brain that are specialized for processing different types of information, right? We have visual information that's processed by one part of your brain, the occipital cortex. You have olfactory information that's processed by olfactory areas in the brain. But when you form a memory, that's a very rich experience. You're going to remember what you tasted, what you smelled, what happened, what things looked like, what music was playing, where you were, when it happened. And all of those types of information are processed by really highly specialized brain areas in other places that are distributed throughout the brain. Now, the hippocampus has to glue all those different types of information together into a coherent memory of an event, like what happened, where did it happen, when did it happen. So the hippocampus gets transmitted all this different type of information from all these different brain areas, and it's going to put that all together into a stored memory. So theta rhythms are really important for that process because they are coordinating the timing of the different inputs that the hippocampus is receiving, transmitting these different types of information. And so I'm going to show you in this very, very, very simplified cartoon, if you can imagine somebody is thinking, where did I put my keys? So there's the what, it's the keys, the where. And so now let's imagine that there's a neuron that represents the living room. And there is a neuron that represents the table. And there's a neuron that represents the keys. And they all fire together, coordinated by these theta cycles. They all get activated together. Then that will help us remember, oh, I left my keys on the table in the living room. Now, obviously, this is a very highly, overly simplified example, but just to give you the basic gist. But what we can actually do, and what we do in my lab, is that we can, this is a cartoon example. I didn't do an experiment where I was able to actually record from somebody who's, remembering where they left their keys, because we can't implant electrodes and record these single neurons from a normal human. They're not going to want us to do that, right? But we have our friends, the rats, and they're the really smart animals, and they're very easy to train, and they can recover from this surgery. They're a very hardy animal. They can be up and going about their business again the next day. And they're quite happy to learn lots of new things as long as you give them sugary treats. They love Fruit Loops. They love vanilla cookies. And then we can record from these individual neurons and see how they are storing memories. Obviously in a simplified way because this is a rat, but nonetheless. So now if you imagine in the rat world, this rat is having an experience that's very salient to them. They have to learn, where am I going to find some yummy food that I like? So they can be in a maze, and there's all these different paths they can take, but they can learn, okay, in this maze, if I go to this place, this is where I'm going to find the really delicious food. And so they will store a memory of all the locations on that maze that they have to pass through to get there. And you can see the memory will be stored in the activation patterns. These are those spikes that I told you about at the very beginning of the talk, these little spikes of electricity coming from these different cells. The cells are color-coded here, different colors. And you can imagine this is like a Morse code. Each cell is coding part of that memory, and they're all getting coordinated in this specific sequence that is going to allow the rat to store a memory of that path in the maze that it takes to get a reward. Now, something that's really interesting is later, when the path that the rat took to get that reward is remembered, it's actually a memory representation that's compressed in time, and it's also much less rich in detail. But if you think about it, so here would be an example now when the rat, after they learned that memory representation of that path to the reward, now they're going to retrieve their memory of where that reward is and how they get there. So you can see the same basic pattern is the same. It's the same sequence of cells getting activated, but there's that Morse code is compressed. It's much less detail. It's just like a single spike as opposed to that bop, 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 and it's compressed in time. And this makes sense because if you think about it, when you retrieve a memory of an experience you had, you don't act it out again in real time, right? It becomes kind of more instantaneous. You remember what happened, but much more, you're retrieving it much quickly. And also a lot of the detail in the original experience is lost. Now you just kind of have the basic gist of the event. Okay, so the really cool thing that we can do in the rats is we can record from many, many, many different neurons in the hippocampus, and then we can decode their activity. So again, I'm going to show you a very simplified cartoon, and then for those who are interested, I'm going to get a little bit more technical at the very end and show you the real thing, the detailed part, but if it's too scientific or too complicated for you, just feel free to just, space out for a second or look at your phone. It'll be over quickly. But in this cartoon, I'm going to teach you the basic gist part of what you need to know. If you can imagine, you're going to record brain activity when somebody is forming a memory of an experience. In this case, maybe this woman is having a visit with her granddaughter. And then here is the pattern of spikes from all of those neurons that are firing together to store a memory of that experience. They're all representing different aspects of the experience, like where it happened, the smells that were present, the face of their granddaughter, et cetera, et cetera. And then we can then feed that into a computer, and we can decipher those neural codes that are representing that stored memory. And then later, we can reconstruct the memory representation again. This is very, very overly simplified, but here you can imagine if this is the pattern of spikes from a lot of neurons, this is only showing, less than 10, but in reality you need many more than that. But this is the pattern of activity that represents the memory of the granddaughter. And then there's a different pattern of activity that represents the memory of the daughter. And then when you want to decode the activity, you record a pattern of activity in that same group of cells and see which memory does it match with, right? This pattern, it looks more like the granddaughter memory. So then you can decipher or decode that activity and say, okay, right now the brain is retrieving the memory of the experience with the granddaughter. Okay, so now we're going to come back to the rats. That's just to give you a cartoon, sort of overly simplified view of the technique. But now we come back to the rats, and here we have a similar thing. This is the rat forming the memory as they have that experience of taking that path through the maze to go get their piece of cheese, and then later, when they want to recall that memory of, okay, what's the way that I go to get my cheese? Then they can emit that same sequence of cells can get reactivated again. And so we can record spikes at a given point in time from our population of cells, and we can use this technique called Bayesian decoding, where we can say, what is the probability of the rat representing a particular memory, in this case, a particular location, given the pattern of spikes that we observed that are shown here N. So you can see at this point in time, the pattern of spikes is what is outlined in this orange box here. And we can solve that using Bayes' rule. Now, Bayes' rule, this is actually something that, have any of you heard of Bayes' rule before? Yeah, you can use this for a lot of things, right? You can start using this, it's like making decisions about whether you're going to go out tonight or something, like what's the risk of things happen, right? It's going to tell you like a lot of different things. What's the probability of you're going to get the outcome that you want? So this same sort of Bayes rule, we can apply it to these spike trains. And so here's Bayes' rule over here on the left. And we say, what is the probability of spiking in all of the different locations? What's the probability of the rat being in a particular location? And then in the denominator, we just have a normalizing constant because the entire thing has to add up to 1. Presumably, the rat has to be at some location or some location. It can't just be out in outer space. And so this probability of spiking in every location, we get from a training, we call it a training data set. So we have an earlier time where we have the rat do the activity, and so we can see how it represents the memory of going through that particular path in the maze. And so then we can use that training data set to plug it in here to decode the activity that we pick up later at this point in time here. Now, when we do this, we see that if a rat is learning to go to a particular location where they are going to find a reward, early in the training, before they know where they haven't learned yet where they're going to get that yummy piece of cheese, what I'm showing you here, these warm colors are the decoded representation, and this white line is the animal's actual trajectory, and then this red dotted horizontal line is the reward location. So location on a track is shown here in this vertical axis, and this horizontal axis here is across time. So as the animal is approaching that reward location, but they haven't yet learned that there's something special about that location, you see that the decoded representation is just very closely matching their actual location. Now, for those eagle-eyed people in the audience, you might see it flickering off and on, off and on. That warm color is coming like these ticks, right? Like a warm, you see it like a flickering, like on, off, on. Here you can see it, on, off, on. That's the theta coordination, because the cells are being coordinated by the theta rhythms. So when you see that warm color, that's when all the cells are being activated in sync at that portion of the theta cycle where they're getting activated, okay? Now, I know this might be getting a little technical for some of you. I promise it's almost over. Now here, on the last trial, when they have learned where that reward location is, they start the activity starts to retrieve the memory of the path to the location before the animal actually gets there. And that's what you can see here. As the animal is approaching the location, when they're far away, the population of cells is just representing the experience that's actually happening. It's matching the animal's position, just their ongoing trajectory. But as they start to approach that location, they remember, oh, that's where I want to go. That's where I got that yummy piece of cheese. I want to go back there. And you can see that the representation, the memory, is retrieved. And you see it sweeping ahead to that reward location, sweeping ahead to that reward location over and over periodically. And again, that flickering, that's the theta coordination. Because now here is a more close-up view of it. Now this is on a correct trial when the animal had to navigate to a location in the maze, and if they went to the right location, they would get a reward. This is after they had learned it, and you can see again now the white line is showing their actual trajectory approaching that location in the maze, and then this warm colors is showing what that group of cells, that coordinated group of cells, what memory is being retrieved. And you can see that as they're approaching that location where they're going to get the reward, the memory of that location's heading to that reward site is retrieved as they are approaching it. And that's happening over and over until they get there. Now, what's interesting is sometimes rats forget, right? Just like all of us, they get distracted or they're not paying attention and they make a mistake. So we can also look at the times when their memory fails. And when we do that, this is an example, we see that we're not seeing the memory getting retrieved properly at those times. In this case here, you see the white line shows the rat's actual trajectory, and these warm colors are showing the memory that's getting retrieved, and it's just matching the actual location where the rat is, but it's not remembering, hey, I need to take this path on the maze to get my piece of cheese. Okay. So I hope that I've convinced you today that brain rhythms are very important for coordinating interactions among large groups of neurons, and especially of interest to my lab that this is occurring during episodic memory encoding. And one thing that I didn't tell you, but I just want to mention in case anyone is interested in asking about it, is that EEG rhythms are disrupted in many different learning disabilities and memory disorders, including autism spectrum disorders and Alzheimer's disease. So discoveries about brain rhythms may lead to novel therapies for diseases and disorders. And so with that, I would like to transition over to the Q&A portion of our talk. Do you want me to put back up the, I'll put back up the QR code in a second if you want to do questions on the app. You're also welcome to raise your hand, as Dr. Mock said. But it's my great pleasure to welcome my panelists who are also going to help answer your questions today. Dr. Laura Fonken, who is my colleague in the interdisciplinary neuroscience program here at UT Austin. She is also a faculty member in the College of Pharmacy. And she is an expert on circadian rhythms, which I didn't talk to you about today, but are really fascinating, very slow type of rhythm compared to the rhythms that I told you about today. But these are the rhythms that are going on in your body that are matching up to your sleep and wake cycles or cycles of day and night, however you want to think about it, those roughly 24-hour cycles that we all experience in our daily lives. And then it's also my great pleasure to welcome one of our top PhD students here at UT Austin, Isabel Moreno. She is in Dr. Kevin Kumar's lab, and Dr. Kumar is a neurosurgeon who studies, among other things, patients who have epilepsy, which is a type of disorder when rhythms go awry. And many of these patients actually undergo surgeries that are analogous to the type of surgery that I was talking about that the rats in my lab undergo. And also, something that is very near and dear to both Isabel and me is our love for dance. I think we both have a love for brain rhythms because we just love rhythms in general. My main hobby is dancing. And Isabel is actually a trained professional dancer, part of a dance company here in Austin, in addition to being a PhD student in our neuroscience program. So I'd like to welcome both of you up here. Okay. Let me turn this one off. Does everybody have the QR code? So I'm going to switch over to the other. So maybe I can just get things started while you're all thinking of your questions. Dr. Fonken, would you tell us a little bit about circadian rhythms and how important they are for brain health?
Yeah, I'd love to talk about rhythms, but as Laura mentioned,
Circadian rhythms.
Yes.
Sorry, that's what I meant to say.
Yeah, but I study them on a different scale than Dr. Colgin. So I'm really interested in the 24-hour fluctuations that take place in our body. And circadian rhythms are coordinated by a different brain structure than what Laura was talking about. They're coordinated by a brain structure in the hypothalamus, so right at the base of the brain, a nucleus there called the suprachiasmatic nucleus. And what that brain structure does is it receives the information about lighting environment from your retina, and then it regulates these 24-hour rhythms throughout your body. So you can kind of think of the suprachiasmatic nucleus as being a conductor of an orchestra, and you have all of these clocks that are represented throughout your body that maybe are the different sections of an orchestra that get coordinated by that suprachiasmatic nucleus. So you have 24-hour rhythms not just in your brain, but also in your liver and in your intestines. And synchrony between all of those different structures or all of your different organs is important for health. And when you disrupt your circadian system, so doing things like shift work, but also doing things like maybe staying up late at night, scrolling on your cell phone, that can lead to disruption of those rhythms or desynchrony between clocks in our body and lead to pathological health outcomes. And that's what we're interested in my lab here at UT.
Right. Oh, yes, go ahead. Could someone bring a micro? Oh, sorry.
Hello.
We'll bring one down to you in a sec. Sorry. Go ahead.
Thank you for the lecture. My name is Michael. As a musician, is it common to have theta, alpha, beta, gamma happening at the same time? And also, the range of the gamma rhythm was a lot bigger, like 120 hertz. So if you could talk a little bit more about that, but thank you.
Yeah, that's a great question. I love that question. Yes, the rhythms happen at the same time, and they're actually interacting with each other. So for example, theta rhythms are slower, and there'll be a particular phase of the theta where the amplitude of gamma will get very strong. And even just to give you a more simplistic answer, there's a lot of other rhythms in our body that I didn't talk about today, and some we're all very familiar with. Your heartbeat, your breathing rhythm, and also your your stomach has a rhythm. And those rhythms are all coordinated with each other as well. So there's a particular phase, what's called a phase-phase relationship. So even though one is faster, they're coordinated. It's just that it would be easier if I had a figure out to show this, but there is a phase when there's some kind of shared mechanism of generation. They're somehow able to be connected to each other so that, there's constantly different rhythms going on at the same time, but they are interacting with each other and coordinating with each other. Great question. Sometimes there's complicated relationships. So using the circadian rhythm as an example, right? Like if you looked at circadian rhythms, you could say that those are actually phase amplitude coupled with the slow oscillation that occurs during sleep. So you would see that at the phase of the circadian rhythm that corresponds to nighttime, the amplitude or the magnitude of the slow oscillations that occur during sleep are going to be larger. So things like that. So there's all these interrelationships that are super interesting. And if you're interested in like the mind-body connection, it's, like Dr. Fonken was talking about, if your circadian rhythms go out of whack, that messes up a lot of other things in your body because they're all connected with each other and all in sync with each other. Yeah, okay.
Okay, first question. Dr. Fonken, this is a personal one. Are you related to Dr. Gerald Fonken, who was the vice provost here at UT and a great professor?
Yes, that was my grandfather. So, yeah. And he used to work in this building. He was an organic chemist.
He was awesome. Now, getting back to circadian rhythms, can you explain to us why springing forward and falling back that we do twice a year, why we need to stop this?
Yes, always a controversial topic. So we have these natural 24-hour fluctuations in our body that happen, but our circadian system is also very responsive to the light-dark cycle. So these 24-hour oscillations in our body, they would take place even if we didn't have a light-dark cycle. So they're endogenously generated internally. But the circadian system evolved to be responsive to environmental cues. So our circadian rhythms are about, but not exactly 24 hours because evolution isn't perfect. So some cells might have a 23.8 hour rhythm and others a 24.2 hour rhythm, or rhythms between different individuals might be slightly different, like morning larks and night owls might have slightly different rhythms. So it's important that that system can respond to the light-dark cycle. And in terms of evolutionary history, that was primarily through sun and sunlight and moonlight at night, so darkness during the night. And that bright light in the morning, that sunlight would help resynchronize our circadian system every day so that we stayed on a nice 24-hour day. We don't do that anymore now, so we use a lot of electrical lighting and have light at different times of day. And when we do something like daylight savings time, we're shifting that clock. And when we shift the clock, there's associations that are shown between things like increases in car accidents right after daylight savings time, because our circadian system is really important for coordinating things like learning and memory. And when you shift those rhythms, you can disrupt some of those processes. But an important plug, whenever it comes up with the legislature level, there's always this idea that we should go into permanent daylight savings time. But that's a very bad idea. So it's really important that we get this early morning light exposure, so we don't want to shift that light exposure later. It's important for things like safety for children who are riding buses to school, things like that. So even though people like the idea of sleeping in, it's actually not the healthier choice.
So we're going to go back and forth a little bit between the app and the audience. So we have one that came in through the app, and I'm going to direct this one for Isabel because I have a personal interest in this as well. This question says, what kind of dance do you think is best for the brain health of a middle-aged woman and why?
Okay, I think that the best type of dance is honestly just whatever dance you can get into. I think it's a lot about consistency, it's about showing up, it's about doing something and enjoying it. A big benefit to dance is like the social aspect, the mental health, and it's like something that a lot of people, including Dr. Colgin and I, derive a lot of joy from. Some things that I recommend people get into, like if you've never done dance before, tap is a popular choice. There's a ton of places in Austin that do it. And then also like line dancing is a really popular choice. And those tend to be sort of like beginner friendly, simple, but you still get to like work your brain, learn choreography, all of the benefits.
I have a question. Does everyone have the same circadian rhythm, or does that vary among different people?
Yeah, so people's circadian rhythms can vary slightly. And sometimes there's genetic mutations that can lead to more substantial shifts in circadian rhythms. as I was mentioning, your circadian systems evolved to be kind of that about 24 hours, but it's not 100% precise. So some people can have slightly shorter, some people can have slightly longer. The circadian rhythms also change over the lifespan. So you might see, for example, earlier morning wake-ups in very young children, and then that shifts in adolescence and teen years to later. So you do see changes between individuals in circadian rhythms.
Change it? I mean, so if someone is used to waking up late all the time, is that natural circadian rhythm for that person, or are we actually changing it?
Yeah, it can be a natural circadian rhythm. The problems arise when someone has, for example, a very long circadian rhythm, so it's maybe farther away from that 24-hour point. And eventually what can happen is that they drift away from the regular light-dark cycle, and that can lead to types of sleep disorders, for example, and be problematic for health and well-being.
Early on, you had mentioned the matchup between the theta cycle and the number of syllables in speech. And there's a perception that some of us have that some, for example, Spanish speakers have a faster rate of speaking. Is there a difference among people or ethnic groups in terms of the rate of their theta cycles?
So the good thing about the theta rhythm is that it's actually a bit flexible in its timing, and it will get what we call entrained by what it is that you're paying attention to. So, but it has its limits. So if you're paying attention to somebody who's speaking more quickly, your theta rhythm will be able to adjust to be faster. And if, like for the rat, as the example to bring it back to the rat, if the rat is running more quickly through a sequence of locations, its theta rhythm will adjust to be faster so that it can take in the changing visual information that's changing more quickly. So there is some flexibility in the timing of the theta rhythms. So I don't think that it would be so much of differences in the timing between individuals as it would be differences in the timing depending on how the stimuli that you're paying attention to are arriving. Now, I will say that there's differences in the timing across species, and that's thought to be because, like, as humans, we have big brains compared to a rat, right? So for things to be synced up from this part of the brain to this part of the brain, that's a long distance, and that there is a delay you need to be able to tolerate. So the theta rhythm in humans is slower than it is in a rat, but either way, there is still a range in the timing, so it can speed up and slow down to adjust for things that, like you're talking about. But if things get too fast, then it can't adjust that much, like it's within a range. Yeah. Oh, yes.
I have several super specific questions that I had written down, but I'm going to choose to ask the one that seems the most far-fetched. If we were able to provide a computer with a large enough training data set on, say, human spikes, human neural spikes data, would we be able to do a similar type of neuronal decoding that allows you to see what the rat is thinking about in a way to combat barriers of people with speech disorders who can't verbalize what they're thinking or imagining?
So here I'm gonna put a plug in for the Brain Initiative, which is public funding for scientific research, because this exact thing that you're talking about is actually happening. So, for example, Eddie Chang at the University of California, San Francisco, he is a neurosurgeon, and they are able to implant electrodes in patients' brains who have lost the ability to speak. and they can think about what it is they want to say and decode it, and then feed that into a voice box that can restore speech for these patients. Research like this is happening right here at UT Austin and Texas Robotics. There's brain-machine interfaces to recover function for motor movement in patients. So patients can imagine how they want to move a limb that they may have lost the ability to control. And our amazing scientists here, right here in Austin, at UT Austin and Texas Robotics, are developing robotic prosthetics so that the patient can just think about what they want to do and they can recover function in that way. So yes, and this all started out as basic research in animal models and is now leading to improvement of lives for many patients. Yeah, it's really exciting.
That's amazing. So
I know, isn't it?
Sounds
so
like
cool?
it's too late for me to get my name on that paper.
Well, there's always, it's constantly improving. So yeah, there's still opportunities. Texas Robotics, yeah.
Hi. I wanted to ask about brain rhythms with respect to relaxation and recovery, specifically sound baths. Are you familiar with the idea of a sound bath?
I am really sorry, but
No,
I'm not.
I can explain. Okay.
Oh, okay.
It's
the gong and yoga.
with the
Okay,
gong,
okay.
and the singing, the Tibetan singing bowls and everything.
Okay. I didn't know it was called that. I'm sorry.
Yeah, I'm just curious because I've been through one. It was incredibly relaxing. Sometimes they can also mess them up if you don't go to the right person. But I'm just curious, kind of, if you know how that would work and what is happening in the brain and how that is able to relax someone so much.
So, and please, someone else feel free to jump in. I don't actually know how those particular sounds evoke relaxation, but when you are in a relaxed state, there are certainly changes in your brain rhythms. For example, those alpha rhythms that I talked about that allow you to focus your thoughts inward and sort of tune out distractions in the world around you. But why those particular sounds are likely to put us into that state, do you know more about it, Isabel?
I know very little about it, but from what I understand, these sounds, the reason they're helpful is because they are at a specific frequency, and I imagine that is why they are aligning with some of the relaxation brain rhythms that we see.
Oh, sorry, go ahead.
How are brain rhythms and just memory retrieval, how does that work? Or how is it affected when someone has a false memory? Is it going to follow that same path that we see? Because to that person, it's a real memory, or how does that work?
So actually, people do think that the storage of false memories can be very similar to the storage of real memories. So one mechanism for storing false memories is that every time we retrieve a memory representation, we do something called reconsolidation. We reconsolidate that memory. But that makes the original memory vulnerable to disruption. So sometimes if you remembered it incorrectly, or something was suggested to you, or you were at the exact same time having an experience that was really similar and was activating part of the same ensemble of cells, then it can actually degrade the original memory representation and make it lose some of its accuracy because either the new memory interferes with it, or something got suggested and then you thought that it was part of that old memory when you retrieved it later, but it actually wasn't. So it is, in some ways it's a separate process, but a lot of the same way of consolidating and retrieving it is similar. So it is really something for us to be aware of, like if we're having those arguments with our sibling about, you're trying to remember some family vacation you went on and, you have different memories of it, it's a lot of times happening, right? And it could be that just a lot of interference when you're retrieving memories and they become just vulnerable to being degraded, basically. So the more we can, nowadays, right, we can take pictures and document things, but our own memory, it's not perfect. Over here, yeah.
What differences would you expect to see in the theta waves in a healthy adolescent brain versus a healthy geriatric brain?
Well, so I don't know that much about, by adolescence, the theta rhythms should be pretty well developed. And in a healthy aged brain, there might be a little bit of loss of the strength of theta, but it should actually still be going strong. In healthy aging, just throughout our lifespan, we are all losing some of those connections between our cells, and we are even losing cells, even with normal healthy aging. But the theta rhythm is a population activity, so it's going to take a lot to make it go away completely. So just normal aging, you should still be good.
So you mentioned that the brain prefers sensory information at the theta rhythm, but is there any benefit to direct stimulation at different theta or other rhythms on the brain, using electrical stimulation to replicate or stimulate at those rhythms?
So I can take that. Yeah, there is thought to be. So people are doing a so-called transcranial stimulation at theta burst stimulation, which is thought to be the optimal pattern for strengthening memories. So these type of things are in clinical trials, and to my knowledge, it looks promising, actually. So yeah, great question.
I am the mother of an individual who had the surgery that your rats had. He has autism and epilepsy. So can you describe a little bit of the theta waves in individuals that have the combination of both?
So I might actually turn this over to Isabel if you're comfortable, because she works in Kevin Kumar's lab, who is studying patients exactly undergoing surgery for epilepsy. So, and then I maybe chime in some extra at the end, but please take it.
Yeah, so we have seen people with this surgery, and really the kind of information that we're looking for is sort of the abnormal synchrony. So like we're specifically, those electrodes are in there to see when the synchrony is, too much. And at that point it would be something, epilepsy, a seizure, something like that. So I'm not sure how something like autism would impact the theta waves, but to my understanding, as far as epilepsy goes, they have healthy brains until suddenly they don't. And that's sort of what people are trying to target with these epilepsy treatments. But maybe Dr. Colgin can speak on the autism.
So one thing, and I want to say this is what my knowledge is primarily from what we are hypothesizing from animal models, because that is more my area of expertise. But it looks like there is a loss of the cells become almost sort of overly excited. And so if you noticed in those diagrams of the theta rhythms, there's a portion of the wave where they're active and then they go quiet. And that's what's really syncing them up. And that portion where they go quiet, that's a really important function of theta 2 that I didn't get into, because if you think about when you're paying attention to the world around you, things are constantly changing. So you don't want to get stuck. You're retrieving a memory, you're predicting something, but maybe that prediction is wrong. So you want to reset it and update it with the sensory information on a regular basis. And so the theta is providing a mechanism for doing that. And what might be happening, and of course there's multiple different kinds of genetic causes and underlying causes of autism spectrum disorders. But what might be happening in some of these cases, and it also aligns with epilepsy, is that there is actually kind of too much excitation in the cells. And so they're not like properly aligned with that one portion of the wave. And then that can lead to the types of activity that Isabel was talking about, these pathological epileptiform events that are actually where cells are just out of control, excited. They're not inhibited enough. It can possibly also, we think that it's associated with some of the experiences that individuals with autism spectrum disorders have, where they're so hypersensitive to stimuli. Their loud noises or somebody touching them might be actually irritating because it's like too much. It's not getting filtered out properly. So the theta rhythms are there, but the activation of the cells are not properly timed to the theta rhythms in at least some variants of autism spectrum disorders. That's what the evidence points toward.
Is there a relation between mental health and any of the brain rhythms?
Yeah, I mean there definitely is. So a lot of mental health disorders show abnormal brain rhythms. There's actually one of the reasons why the EEG is such a powerful tool, diagnostic tool in neurology. We actually have one of our neurology colleagues here. I might actually call on Dr. Schwartz if he wants to weigh in on any of this. He's like, no, please leave me alone. But it's a very powerful diagnostic tool because it's often used as, I'm not a neurologist, so I don't want to overspeak, but let's say, for example, if somebody is possibly having attention deficit to hyperactivity disorder, and you see those patterns of abnormal EEG that are associated with that, then that can lead to a better diagnosis. Also, the way that the brain is responding to sensory stimuli, you can pick that up with the EEG. Is it responding normally? I didn't talk about gamma rhythms. I know there was a question about them. But gamma rhythms are a type of rhythm that are associated with processing sensory stimuli, but on a very fast time scale that's outside of our conscious perception. And they're very local. So they're really important for kind of coordinating localized groups of neurons that are specialized for like a specific function. And if sensory information isn't being processed correctly, then there's oftentimes disturbances in those rhythms, like, for example, in schizophrenia or other mental disorders. So yeah, really, I mean, the list could go on and on. It's because brain rhythms do reflect the coordinated activity across large populations of cells, they're actually good metrics for detecting impairments, because if you're just sampling individual cells, maybe one cell is healthy, but other ones are not, then it's really dependent upon which cell you happen to sample from. But the EEG reflects the activity of the entire network, and so it's kind of like, what is that saying? For the want of a nail, the shoe was lost. For the want of the shoe, the horse was lost. For the want of the horse, the battle was lost. For the want of the battle, the kingdom was lost. It's like that one little thing can have devastating effects when you go up to the higher level, and that higher level is the measurement of the brain rhythms. I did, but you look like you didn't want to. No, yes, please, Dr. Schwartz, speak up.
No, I would just add that the analysis of the EEG has gotten very sophisticated.
Bill.
Professor of neurology who actually has expertise in humans, not rats.
Expertise in humans, no. Just personal expertise as a human. No, I would just add to what Laura said, that the analysis of EEG has really gotten very sophisticated now on an experimental, investigational basis. that's beyond sort of in the clinic where the EEG is used for epilepsy and for sleep studies. I mean, now they have, for example, high density EEG with 256, something like that, electrodes. And so learning about sort of not only these waves and the interaction of these waves in time, but also in space, so that within lobes of the brain, the interactions as well. So all of this is exciting, trying to sort out sort of the mechanisms and where things might go wrong.
Yes, hi. Go ahead.
When you were asked about the false memories, the mechanism that you explained, is it similar to the way déjà vu works?
I don't actually know how déjà vu works. Do either of you know? I don't know. Oh, Dr. Mock, do you know anything about how déjà vu works? Okay, I can tell you one thing. This is very anecdotal. So as scientists, we're more about like designing experiments with controls and things like this. But there has been some deep brain stimulation studies where they have stimulated pathways to the hippocampus, and the patients state that they have a feeling of a déjà vu. So that's about as much as I can tell you. I think that the hippocampus does think it's retrieving something familiar, but why that just happens by coincidence, it's probably just a random activation that comes up.
Hi, so I kind of have two questions now, because as an addendum to that, I don't know if you would have any more insight on this than with déjà vu, but now I'm also thinking about jamais vu, which is when you've done something so many times, but now you feel like you're doing it for the first time. Kind of like if you try to think about riding a bike a little too hard when it's become motor memory. But what I wanted to actually ask about was, I think it was really interesting to learn about all the variables that are conducive to facilitating the theta rhythms and thus some long-term and episodic memories, such as like playing videos at their intended speed. And in thinking of that, I'm wondering how many of those variables are similar to the variables that are conducive to aiding the gamma rhythms, for things like performance. And so, for instance, I'm thinking about things like speed cubing and trying to get like faster and faster times.
What, speed cubing?
Yeah, like the sport where they solve Rubik's cubes in seconds.
Oh, okay. So a couple things. So those types of procedural memories, like solving a Rubik's cube, that's not the type of that the hippocampus wouldn't be involved in that type of memory. And we have Dr. Mock here, who may want to jump in and talk about those sort of coordinated motor memories and the brain systems that are important for that. But your question about the theta and gamma rhythms, this comes back to the question that we had earlier. They are certainly interrelated with each other, and many of the same behaviors that increase the theta rhythms, they also increase the gamma rhythms, because I was talking about the different types of relationships. I talked about phase amplitude coupling, but there's also amplitude coupling, which is when theta rhythms get larger, gamma rhythms get larger as well. So they are working together. They're doing different things because, like I said, theta is you're interacting with the world around you. That's something that you're sort of consciously aware of. Gamma rhythms are doing more things that they're very fast. It's outside of the range of our conscious perception. They're doing things that are faster than we are aware of. So they have a different role. So they can be, for example, filtering out irrelevant things that are happening. We're not necessarily actively, consciously doing that, but your brain is doing that automatically, right? Like if I tell you, oh, listen, do you hear the noise of the fan of the projector? Yes, you hear it. But the whole time you've been here, right, your brain has just been filtering that out. So gamma rhythms might be more involved in those sort of things that feel automatic because they're just happening so quickly.
Is that related to like declarative versus non-declarative?
Not necessarily, because no, gamma rhythms can still, they have a role in episodic memory and declarative memory. It's just there's aspects of that memory encoding that need to be done faster than what the theta rhythm can do. Yeah.
I've got a question. Whoops, okay. As I was watching the rat go through the cheese, going forward, it reminded me of when I used to study for final exams. So my question is the role of memory and repetition in learning.
Yeah, no, I mean, repetition is obviously great for learning, right? That's a way for us to remember things, is to repeat it over and over. And that is actually the case both for declarative memories, right? Like, you know, I didn't know where this room was. This is actually the first time that I've been here. But I teach in this building, and I don't have to really, I can just, you know, automatically remember where my classroom is because I go there every Monday and every Wednesday. But this room, you know, maybe the next time I would try to come here, I would have to write down the number again and, you know, follow the signs to get here. But if I repeat it over and over, right, then I will definitely remember it automatically. You know, I don't have to follow the signs. And what's interesting is that the other types of learning that we were talking about, the more procedural types of learning, which, you know, dance is also something that is in that category. You know, the more you practice, the better you know that as well, right? Like if you're learning choreography. So that repetition is important for, yeah, all types of learning. Unless it's something bad, then you don't want to repeat it.
Hi. You had an amazing talk, by the way, first of all.
Oh, thank you.
But also, I wanted to add something on the fact that you were saying something like memory is hard to read in humans, right? Because, you know, we don't have specific enough EEGs for that. But can you talk a little bit about, you know, when those memories get sensitized, like in PTSD, do you think that there's a way to read that in an EEG?
Well, and again, we have Dr. Mock here, who is an expert on this probably more than me. But to my knowledge, that would be something that would be difficult to measure at the level of the EEG. But something that is really, really fascinating about those type of memories and was discovered by our colleague, Michael Drew, right here at UT Austin, is that when you're actually having a traumatic memory like that, there is the representation of that original memory, but if you want to extinguish that, there's a separate memory representation for that extinction experience, and it can compete with the original memory representation and kind of win and suppress it. So there are really, you know, ways hopefully to overcome those traumatic memories. And Dr. Mock, would you like to add to that?
Yeah, it's an example of how different kinds of memory are mediated by different parts of the brain. So that's not hippocampus. Fear memories are in the amygdala, and it has its own separate rules and its own. Are there rhythms in the amygdala?
There are, but it's controversial because, okay, I mean, this is getting really into like the insider topic, but to measure the rhythms, when you're measuring things extracellularly, the alignment of the cells, the way the cells are arranged is really important because, yeah, you have synchronous activity among the cells, but the currents need to be flowing in the same direction so that they will sum up nicely in a way that can be recorded. And so in areas like the amygdala that are arranged in nuclei, as we would say, it's just a mass of cells. And so you can have, you know, the currents are not going to necessarily all be flowing in the same direction. So in those type of brain areas, there's often people that are able to record the brain rhythms in the area, and other people don't believe they are there. But I think that Dr. Mock would agree with me. They do have theta rhythms in the amygdala. They're harder to record, I think, in any of those brain areas that have The neocortex is arranged in layers. That means all the dendrites are like the antennas of the cells, which we call dendrites, are all in line with each other. So you have this really beautiful summation of currents, and you can record brain rhythms beautifully with extracellular recording electrodes. But yeah. But I don't want to be disrespectful to those people that don't believe in the amygdala reports of theta.
Is this on? Okay. I think earlier you mentioned some work that was done in reinforcing or strengthening memories. I was wondering what the current research is into hyperthymesia, or the highly superior autobiographical memory, and why is it only 20 people in the world have it? It just seems like, or is there anybody knows anything about research being done on that?
So I always get this question, so I need to start researching this. I know there are people that are studying these individuals, and in fact, there are people at the University of California, Irvine. So if you're interested in this, they often have public talks. They're online. And I would encourage you to go to their website, the Center for the Neurobiology of Learning and Memory. That's where I did my PhD. And I know that there are researchers there that are specifically studying these individuals that remember every single thing. But I have to be totally honest, I have not kept up to date with this research. I don't have knowledge about it. As you mentioned, it's a really small sample of individuals that have this type of skill. And so it's kind of a niche area that I myself am unfamiliar with. I apologize. But I know that there's really innovative research happening. So I encourage you, do a search, University of California at Irvine, autobiographical memory, and you might even find a public lecture posted online about it.
I was curious about I'm picturing there was the guy that was up here talking about, are the brain rhythms like music? So when you look at something like autism spectrum disorder or Alzheimer's or Parkinson's, and there's maybe that disconnect of the rhythms that are going on. And so we know movement is so important to get people organized, but then I think about what happens in public school a lot, where recess becomes less and less, so less movement, less organization. That's just one thought. But what is it about movement that is so important? Is it touching upon different areas in the brain, like this part's responsible for this, and then all of a sudden it's coming together and organizing? That's my question.
Isabel, do you want to take that one?
Yeah, so something that is really beneficial as far as like exercise and coordinated movement is that it connects your whole brain. So it really has like this wide activation. And there have been studies showing that like people who dance a lot have higher amounts of white matter in their brain. So that's like the connective part of your brain. Their brains are able to sort of connect all the way across. And then that's sort of the rationale for things like Dance for Parkinson's. It's like a program for people with Parkinson's, and they will, okay, yeah, you're familiar. Yeah, so the benefit for that is that they get this increase in connectivity, and hopefully that can help them manage their symptoms. And it also offers a lot of social benefits. So you can see synchrony in brainwaves while people are dancing together. And when you talk to people anecdotally about these Dance for Parkinson's programs, they'll tell you like, you know, this is the best part of my week. Like, I love coming here. And it's because they get a lot of social connection from it as well.
Hi, we'll stay back at the table here for a second. So I was wondering with circadian rhythms, you know, they are something that's actually, you know, part of us, but and then there's OCD behaviors, which seem to be must be related to some sort of arrhythmia or however. Is there a feedback either between circadian rhythms being disrupted, triggering OCD, or conversely OCD triggering disruptions in circadian rhythms?
Yeah, I'm not as familiar about associations between OCD and circadian rhythms, but disruption of circadian rhythms are implicated in mental health. So, for example, things like shift work are associated with increased risk for anxiety and depression. And then there's been a lot of interest in terms of young individuals with things like screen use late at night, cell phones, how that parallels increasing rates of anxiety in those populations. So there's definitely effects of disrupting rhythms on different types of brain function, but I'm not as familiar in the context of OCD. I don't know if audience member Bill has.
Dr. Schwartz.
So there was a lot of interest in bipolar
Yeah.
disease and some kind of interaction between circadian dysrhythmia.
Yeah.
OCD, I'm not.
Yeah, so there's been studies in bipolar disorder, for example, looking at rhythms in postmortem human tissue and trying to figure out whether there's weaker rhythms in patients with bipolar disorder compared to neurotypical counterparts and research like that.
Well, folks, I'm sorry to say that we're out of time. So please help me thank Laura, and Isabel for